Hello, SEA-PHAGES. Welcome to Direct Isolation and Plaque Assay Week. I'm going to be walking you through the direct isolation and plaque assay protocol, some of the mentalities behind it, why we do what we do, and also like physically how we're going to do it. If you want to follow along, this is going to be in your lab manual, pages 24 to 32 and 35 to 37. If you want to take notes, things like that. Okay, so let's get into it. So let's talk about what we're trying to accomplish, right? We're trying to understand different methods to isolate a phage. There's more than one from a soil sample. We're trying to get more practice with proper aseptic technique, make sure you're understanding when to use it, how to use it appropriately, things like that, so we can directly isolate from our environmental sample. And then again, we're going to be building on our pipetting techniques, knowing when to use which pipettes, and basic aseptic technique. So let's start with what is direct isolation. That's what we're going to be doing in lab. It's protocol 5.2. So what is it, right? So it's essentially a really quick method that we can use to detect the presence of phages from an environmental sample. So we want to be able to know if there are phages in our soil that we're collecting. It provides a snapshot of all phages that can infect your host, which is going to be, in our case, Gordonia terra. Okay, so phages typically are, number one, they're everywhere. Anywhere there's bacteria, there's going to be phages. But when we're talking specifically with soil, they interact such that they form these electrostatic interactions with soil particles. So part of our direct isolation procedure is to break up those interactions. We're going to be shaking our sample with some liquid media for an hour, and that's going to work to break up those electrostatic interactions so that eventually we're going to get a solution on top in our tubes. That's going to have our phage in solution with the dirt kind of settle to the bottom. Then we're going to filter that, and we're going to produce something called a filtrate, which we will then be using to conduct a plaque assay. Okay, so basically, a direct isolation gives us the ability to detect multiple different types of phages that can infect our host, very important, that can infect our host that are in our soil sample. So this varies from enriched isolation. So while we're not going to be doing this directly, or you're not going to be doing this directly in the lab, it's important that you know the difference between the two. Okay, so essentially they start off very similar, an enriched isolation compared to a direct isolation. We get a filtrate that we produce from our environmental sample, but with a direct isolation, we immediately plate that sample and try to see what phages we have. With an enriched isolation, what we do is we seed that phage filtrate with a culture of our host bacteria and let it incubate for a couple days. So what happens is that the bacteria is going through replication, the phages are going through several cycles of infection and lysis, such that the culture that you produce after those couple of days are through is a sample that has a really high concentration of phages that are very specific to our host when you compare it to your initial sample. So you can see on the right figure of like a before and after, you can see significantly more phages, these little orange guys, in the after, right? So what it does, what enriched isolation is it is essentially a method used to amplify the number of phages from your environmental sample before you start plating and trying to identify those. Okay. One of the drawbacks is that you get less diversity. So you can consider in these, when you're doing an enriched isolation, usually one, maybe sometimes two types of phages. When I say types, I mean like species. Species of phage will be able to outcompete other ones, right? They can infect better, they can replicate faster, and they can lyse a lot quicker than some other ones. So what's going to happen is they will outcompete those other phages for bacteria, such that towards the end of your incubation period, you're probably only going to have one or two different types of phage within that solution, right? So versus a direct isolation, you might get several types of phage you're just able to infect your bacteria. You get more diversity in phages when you do a direct isolation, but you are getting a higher chance of actually seeing something on your plate when it's an enriched sample, just because you have a higher concentration of phages that you know infect that host. So some drawbacks with both, but important for you to know the difference, okay? So this is just a visualization so we can have it clear in our head. Both processes start exactly the same. They start with some soil. We incubate it with some media, break up those electrostatic interactions. We filter that solution until we get something called a filtrate, that is just solution with phage in it. And then that can go two routes. One, direct isolation, which you will be doing. We just add some bacteria, some cop agar, and we immediately plate it to get, try to visualize those phages. With enriched, we let that bacteriophage and bacteria incubate for a couple days until there is a high concentration of phage, and then we plate it. Okay, so if you need a visualization, you're a visual learner, here is a great figure for that. Okay, but let's get into the protocol, right? What we will be physically doing in the lab. Okay, so number one, when you're first coming into lab, some things that you should be doing each time, but since this is your first time, I'll just run through them. Okay, so number one, find an empty lab bench to sit at. Again, we're going to be using two different rooms, 330 and 336. So if you see that 330 is full, there's only one person per lab bench. Just move into 336. Okay, some days you might be in one, some days you might be in the other. It just depends on the schedule. Okay, put on your lab coat. So if you brought your own lab coat, one from home or one that you bought or have used for a different class, you can just put it on, right? If you are borrowing one, you can navigate to an area where we're going to have a bunch of lab coats for you to borrow. So find one, go through them, find one that fits. Again, these have to go below your hip and they have to be able to zip up or button up depending on the lab coat. So make sure it can do that before you claim it as yours. This is going to be one you're going to be using for the whole semester. Okay, so find a good one. We most likely will have, there's going to be a lot from people to borrow from, but if you're coming in to some of the later labs and you're worried about not being able to get one, you can always just purchase one. So what you should be doing every time you're coming into lab is washing your hands and then wiping down your lab area. This is very good given the COVID situation, but the main idea behind that is that you're not bringing in any sort of germs with you to lab that could contaminate your work, and making sure that anything that's left over from the previous class or previous section is also cleaned off from your area. Okay. Every time we're going to have a protocol at the back of the lab room. There's going to have all the materials that you're going to need for your procedure. So navigating back there, getting what you need at the time is the best tactic to do. Okay. And then you can begin your protocol. So let's start off with our soil samples. So you guys should be bringing into lab 3 different soil samples with you to be extracting your phage samples from, right? So these should correspond, they can be labeled however you want, as long as you're able to understand the differences between them and they can correspond to somewhere in your lab notebooks. So in your lab notebooks, make sure again that you have the following information per soil sample. So that's going to be date and time collected, the location, the physical GPS coordinates. If you have an address, you can put that address into a variety of apps and you can get GPS coordinates produced at the end, okay? The depth at which you got that soil from, the moisture content, again, was it muddy, was it dry, sandy, things like that, the temperature outside when you collected it, and then anything novel, anything defining about it. Was it next to anything, like next to a zoo? Was it next to like a field of manure, like something like that, something novel about it, or just interesting, important that you have that. So for each soil sample, you're going to transfer some of it into a test tube until it's approximately 1/3 of the way full, okay? Make sure that your test tubes are very, very well labeled and they should correspond to your soil sample somehow. So for example, if you label them 1, 2, and 3, then you should label your tubes with your name, the date, and then however you were going to distinguish that soil. So I would say, since my initials are MS, I will put MS 121 and soil sample 1 or something like that. Okay. When you're transferring soil into your test tubes, avoid adding anything like debris-wise, like pieces of wood, because they're going to float to the top in our solution. That's going to interfere with some aspects of our experiment. So try not to incorporate any like chunky debris. Let's just go for dirt. Okay. So you're going to be doing this for each soil sample. So you should have 3 test tubes at the end. You're not putting them all in one, 3 separate test tubes, all very appropriately labeled. Okay. And then aseptically, you're going to add some PYCA media to your test tube with your soil. Okay. You're going to add it until it's about 2 to 3 milliliters submerged in that media. Okay. You're going to be using a serological pipette for this. Just because this is everyone's first lab, I'm going to walk you through which pipettes to use. But once you're moving forward, this should be something you should be understanding yourself, right? If you know the measurement of what you're adding, you should be able to critically think, okay, this is the type of pipette I need to use, okay? But you're going to be using a serological one because that is the type of pipette that measures in milliliters and you're adding milliliters. But an important aspect to remember is that you are adding enough media until it's submerged beneath 2 to 3 milliliters. You're not adding 2 to 3 milliliters, you're adding until it's submerged. So typically that's going to be around 7 to 10 milliliters. That's usually the sweet spot for people until it's about 2 to 3 submerged. Okay, but it all depends on the type of soil you have. So don't be blindly adding it, add it pretty slowly. Okay, once you get your media into your soil samples, just invert them a couple times to get it mixed thoroughly, and then give it to your TA, and we're going to put that into a shaking incubator. Okay, the samples are going to stay in the incubator for about an hour, depending on how quickly we can get everyone's in, but we're shooting for about an hour and at 250 RPM. So they're going to be shaking the whole time, kind of again, to break up those electrostatic interactions. So during this time period, what you can do is turn off your flame. You're not going to be needing it for an hour, so why waste ethanol? Just turn it off, just put the cap on, turn it off. And then you can do a bunch of different things. You can prepare materials for the following steps, which I'll explain through. You can start labeling things like that. And you can work on your lab notebook, right? You just did some steps, write that down somewhere. So what goes into your lab notebook, right? There's a whole other PowerPoint that I'm sure your KA has shared with you about what goes into a lab notebook. But just so we're all on the same page, you're going to have the date and the title of your protocol, the purpose and the aims of that particular protocol that you're doing, the procedure, what you actually did, and not what's written in your lab manual. You'll notice, hopefully at this point, that there are certain things that we are doing that vary a little bit differently from your lab manuals, right? So what we actually did. And then the results, analysis, and future plans. This is not something you can do this week because they are dependent on your results. But next week, when you get results, you can include photos of your results, you can interpret them, and then you can talk about what you're doing next. Okay. So all you need for this particular week is going to be your title, right? Direct isolation, plaque assay, the purpose of that protocol to extract phage particles from an environmental sample, soil sample, and then visualize the presence of those phages in your sample of the phages that are able to infect Gordonia terra. And then results, again, you'll get those next week. So after that hour, 45 minutes to an hour period of time, we are going to spin our samples to settle the soil at the bottom and let liquid settle up on top. Okay, we're not going to let it sit on our lab benches for 10 minutes. We're going to just spin it for 5 minutes at 2000 G in a centrifuge to be able to get those two to separate out. Okay, and we will just do this for you. So once this is done, we are moving away from incubation into the filtration steps. So this is all going to be done aseptically, very important, especially because we're going to be working with individual samples and some communal materials. So working aseptically is very important here. Okay, so you are going to take a syringe. This is going to be a sterile syringe, so don't remove it from the packaging until you're absolutely ready for it. And you're going to be working within your aseptic zone. You're going to take your syringe and you're going to extract from this top liquid here. Just place your syringe and extract up about 2 milliliters of your liquid. Okay, and again, if there's any floating debris on top, anything that you might have accidentally added in, avoid that as much as possible, right? Don't try to suck that up if you can avoid it. Once you've sucked that liquid up, you're going to attach a 0.22 micrometer filter to your syringe, kind of here on the left. So this filter is sterile, just like the syringe. So don't take the filter out of the packaging until you're absolutely ready to use it. Okay, a tip that I give students is that you'll notice on the packaging, it comes a little container and a little lid that you can like slide off. You can take the lid off and just leave it really close to your burner. Not too close that it burns, but close. And then when you're ready, you can just screw it on. Okay, so the reason that we use a filter is to filter out any bacteria. Bacteria on average is about 0.5 to 5 micrometers in length. So all bacteria in our sample should be filtered out when we use a 0.22 micrometer filter, right? They're too big, they won't pass through. But phages, they're a lot smaller, they will filter through. The reason that we are using the filter and filtering out bacteria is because we only want the phages to infect one type of bacteria, and that's going to be Gordonia terra. If we are not filtering out other bacteria, we have no way of knowing if the phages that we are growing are infecting the bacteria that we wanted to, Gordonia, or something else, right? We're trying to control any sort of contamination. So you're going to attach the filter, it's a screw-on, Really simple. And then you're going to release the liquid into a microcentrifuge tube. And this is going to be a labeled microcentrifuge tube. And this is now going to be called your filtrate. Okay. You'll notice that when you're pushing through the filter, there's going to be a little bit of resistance, which is normal. But if you're feeling a lot of resistance, then what's happening is likely there's some sort of blockage, usually something with soil, some like a chunk from debris got stuck in your filter and you won't be able to push it through. It's best to just stop, remove the filter, get a new one, attach it, and then push through into your microcentrifuge tube. Okay, don't try to push through too much resistance because it will just like explode out, which we don't want. So you're going to be doing this per sample, right? So this will be per sample. So for every single soil sample you have, you're going to get a new syringe, a new microcentrifuge tube, a new filter. Okay, so at the end, you'll have 3 microcentrifuge tubes filled with approximately 2 milliliters of your liquid, of your phage solution. All of them are going to be labeled appropriately, very important, right? If I come over and mix your 3 tubes around, you need to know which ones are which. Okay, if you drop them on accident and you pick them up, you need to know which one's which. All of these things have happened. Okay, so let's watch all that together.
Direct isolation. Use aseptic technique and work close to a flame while performing this protocol. You will need your environmental sample and liquid media. Add media to your environmental sample until the sample is submerged between 2 to 3 milliliters of liquid. Cap the tube, mix thoroughly, then incubate the sample with vigorous shaking. After shaking, allow the sample to sit undisturbed until most of the particulate matter has settled. You may use a centrifuge to hasten this step. Using a syringe, draw up some of the supernatant. Attach the syringe to a clean filter and collect the filtrate in a clean microcentrifuge tube. This is your filtered direct isolation sample.
So that's it. That is exactly what you guys will be doing. The only exception is you're going to be doing it with an ethanol burner on, all within an aseptic field. Okay, but the protocol is the same. The tools you're going to be using are exactly the same. So rewatch that video. I've linked it below if you want to just watch it a couple times, get familiar with the protocol. But very simple. Hopefully we got that. Okay, so now that we've done that, that was our direct isolation, right? We have isolated some phages into a solution from our environmental sample. Now we have to visualize those phages, which is where the plaque assay comes in. So what a plaque assay does, what it allows you to do, is visually confirm if there are any phages present in your sample, in your environmental sample, right? You can't, looking at your little centrifuge tube, there's no way to know if there's actually phages. We can't, with our human eyes, see little tiny microscopic phages. So we have to do another step, right? So what we do is we mix our host bacteria, Gordonia terra, with our filtrate, and we grow it on a lawn, on agar. And if phages are present and they can infect the bacteria that we are giving it, they'll replicate, they'll kill the host, and then it'll produce something called a plaque. A plaque is a visible clearing. It's usually a perfect circle on the bacterial lawn, very noticeable. And each plaque is the result of a single phage particle infecting and killing that bacteria. Okay, this is not something you will see this week. It takes a couple days for these things to grow, so you'll see them next week as a way to confirm if you do or don't have phages in your sample. Okay, so let's break it down. How are we doing this? So everything you're going to be doing here is going to be aseptic, very important. So you are going to take a micropipette. You are going to transfer 500, all aseptically, 500 microliters of your filtrate into a new culture tube. You're going to label it. Then you're going to add 250 microliters via micropipette of your Gordonia terra bacteria. Make sure that when you're grabbing your bacteria and before you're putting it and aliquoting it into your culture tube, that you shake the tube up a bit. The reason for that is sometimes the bacteria settles. So if you don't shake it, you might not actually be getting bacteria in there. So make sure you're giving it a good shake until everything is incorporated. Usually you'll be able to tell if there's any separation because there's a little tiny pellet at the bottom of the tube. It's just a quick shake and it'll be good. So you're going to be adding that to your culture tube, just a quick pipette up or down or just a quick little swirl to just mix it up a bit. And then you're going to leave that alone for 10 minutes. This is your inoculation step. What happens here is you're essentially giving it 10 minutes for the phages to attach to your bacteria. Okay. The longer you do this to 10 minutes, the more chance of them actually attaching and something growing is going to occur. Okay. So make sure that you are changing tips between each sample, right? You should not be using the same tip for your filtrate and your bacteria. Use different tips between solutions and between samples. Okay, so you're gonna be doing this 3 times for 3 different soil samples, all of which are gonna be labeled. So after that 10-minute inoculation period, we're gonna add top auger. Okay, top auger is molten, so it has to stay in a water bath, 55 degrees Celsius until it's ready to be used, because the second you take it out of that water bath, it's gonna start solidifying, which is not what we want. So don't get it out while you're waiting for your thing to finish inoculating. Get it out when it's done, when you're absolutely ready to use it. Okay. You're going to be using a serological pipette for this, again, because this is in milliliters, not microliters. You're going to transfer 3 milliliters of the top auger into that inoculated solution. Okay. Make sure that when you're getting the auger out of the water bath, it's not too hot. And when I say too hot, if you can touch it and pick it up, then it's not too hot, right? But if you touch it and it's like, oh, that's kind of hot, I don't want to pick it up, that's too hot. And I think you could probably understand why we don't want something that's too hot added to a solution of microorganisms. That's because it will immediately kill it. If you add something that's way too hot for you to touch to something with bacteria and phages, you're going to kill them. But again, if you are able to physically pick it up and hold it, it's not too hot. It's important to work quickly with this, again, because it's going to start solidifying. So as soon as you add it into your culture tube, don't let it sit for any amount of time, right? There's no waiting period. You are immediately pouring it onto a petri dish. Okay, it's going to be a labeled petri dish also. Make sure we're doing that. We're labeling the agar side, and you are pouring the solution into the agar side. And I'll show you a little picture right after the slide to make sure we're getting that. You're going to pour your top auger inoculate solution into your petri dish. You're going to give it a gentle swirl to make sure we're spreading all the agar around. It doesn't have to be too aggressive, very gentle. And you're going to let it sit until that solution is solid. Depends between people. Sometimes that can be 5 minutes, sometimes it's 15 minutes, but just best thing is just don't touch it. Okay, so you're going to be doing this per sample for a total of 3 different plates, all of which are going to be labeled. So labeling is very important. Again, I mentioned this in my previous video, but just as a refresher, we're going to have our name and initials, just so we can recognize you, or you can recognize your own plate, the date, and then the contents, right? So like soil sample 1 plaque assay would be a really good way to label the contents for this particular petri dish, okay? So the agar side is the bottom here, and this is the lid. And you can see that a little bit better here. Agar side, where you can see this like media here, lid. You're going to be labeling on the agar side. Okay, this is also how we store them when they're solid. If we store them with the lid up versus the agar side up, the lid, there's condensation that forms. So if we store it with the lid side up, the condensation drips down into your agar, onto your bacterial lawn, and interferes with how that lawn grows. It's going to interfere with your results. So make sure once they're solid, you flip them and they start agar side up. You're going to tape them together once they're solid. But also, again, you really try to emphasize this. You're going to be pouring your agar inoculate solution onto the agar side here, okay? Not on the lid. I can't tell you how often this happens. Just lift the lid really quickly. Never put the lid down onto your contaminated lab bench. Hold it, pour it onto it, give it a little swirl, close the lid, and leave it alone, okay? But pay close attention to make sure you're not accidentally pouring your solution onto the lid. So let's watch that happen.
Plaque assay. Use aseptic technique and work close to a flame while performing this protocol. Begin by setting up an infection. You will need an aliquot of host bacteria and a sample containing phage. Transfer a small volume of your sample containing phage into the tube containing your host bacteria.
Notice how he's not putting the cap down?
Mix by flicking the tube or rotating the tube at an angle.
Don't invert it because it will come out of the cap. Just a gentle tilt.
Allow the sample to incubate. Once the sample has been incubated, transfer warm, molten top agar to the sample. Then transfer the entire mixture onto an agar plate. Tilt the plate to spread the mixture evenly. Allow the top agar to solidify before moving the plate.
So that's it. So one of the key differences here is that, again, he didn't do this aseptically just for visualizations. You will be doing this all aseptically. Another one is that he used the serological pipette to pour that top agar inoculation solution back into the petri dish. You can do that, or you can just pour it. Both ways work really well. I always encourage pouring just because it's a little bit faster than students figuring out the serological pipette situation, especially for their first time. So either are okay. So that's it for protocol, what exactly you're going to be doing. Obviously, if there are aspects that you didn't really follow along while you're watching this, go back to the slides, go back to parts where I was explaining things, and just rewatch them until you really get a good grasp on this. This lab will take 2 hours, but it's very fast paced, right? To stay on time with everything, especially given the block of incubation time. It's very important that you are able to do this really quickly. So that means coming into lab prepared. So again, if you're struggling with certain concepts, you can read the manual, you can rewatch videos, or you can just look at the slides until you get it. Okay, you can also reach out to me or your TA and we can clarify anything for you. Make sure that if you are not already, that you are signed up for an open lab session to make sure that you're able to come in. If there's any issues with you not being able to sign up or any other things, contact your TA as soon as possible so we can try to figure this out for you. Okay, hope everyone has a great first lab. They're super prepared and excited about isolating their very own phage samples. And hopefully I will see some of you there.